Okay. A couple of things. Housekeeping wise, I'm typically going to be using the Marine PowerPoints. We're going to do mail today. We're getting through as much of it as we can. We got through the whole thing by race. Because I understand that Dr. Robbins was pretty thorough in covering a lot of the repro stuff. So, I mean, I'll give you my spin on what I think is important for you for this exam going forward. So I'll have to look at the exam to see if there's anything I can exclude. And so that's that. If you're, hopefully you can open, those were older dictations. The exam hasn't changed from when I last administered it, at least 2.5 years ago or so. Whenever that was in 23, so that would be 25, whatever that was, a year and a half ago. And so I'm using the same record. I recorded them in 3 sections. If you're having trouble opening them, sometimes it's hard to open them directly from the email. Sometimes you have to download them and open them. That's possible. So you can open it, download it, try to open it. If there's still a problem, let me know, please. And because I found the original source recordings and and I basically reprocessed them on YouTube if there's a problem. And so there's one from 12/1 of that year of 23, 12/3, and 12/5 in order. That should cover the material. Okay, that's out of the way. Now we're ready. And so this is the one I just loaded both of these on there for you, and this is the one we'll be doing. That's here. We're not going to do the female today. We touched on a little bit of female stuff. We take it back here. There's some general reproduction stuff, and then the focus point is the male. I'm trying to focus mostly on physiology. There's going to be some anatomy with it. They come together. To me, to summarize it, the male, the anatomy is the more complex aspect of it. The female, not so much. And the female physiology is much more complex. The male, not so much. The male phys hardly takes any time at all compared to the male, to the female, and to the female physiology. I understand he did the ovarian and the menstrual cycles. That's a real focus point. So I'm going to spend that's Wednesday, and I'll be spending a fair amount of time. And I hope to get everything done by Friday, so we don't have to necessarily be here Monday afternoon, if possible. It's Science Day. If you didn't know that, we got stuff going over there in the atrium. There's some nice presentations that go on. There's undoubtedly food. I'm cooking. Thinking rum cake. I'm thinking pork al pastor tacos, and for our vegan friends, maybe some tofu tacos and stuff like that. I'll go a little crazy over the weekend if I can. Part of me, I have the inevitable issue with a kid, so we'll see what happens. You'll learn this. When you're a parent, you're always a parent, forever. You're stuck. Moral of the story, don't have children. When in doubt, refer back to rule number 1. Okay, what's interesting about this system, obviously we know it does not become active until puberty. And I'm not going to go over the base stuff. Effectively, you have 2 primary structures, testes and ovaries. Those are the prime structures of the reproductive. Everything else is an accessory structure. It doesn't matter whether it's a uterus, whether it's a fallopian tube, whether it's an epididymis. It doesn't matter. They're all accessory structures. These are the 2 primary sites. And they're double duty organs in the sense they are both, effectively, their glandular function or their organ function is both endocrine and exocrine. Exocrine means eggs and sperm because they go through a ductal passageway. Endocrine, testosterone, progesterone, and estrogen. They go directly into the bloodstream. Even though their focal points may be mostly in this area, it doesn't matter. They're like that. And these are just definitional things. I'm not going to ask you about that. Going forward, in many of your, whatever your course is, if you're going into healthcare, a lot of things that you'll see is para and grava. Grava means pregnant, even though you use the term gestation here. Para, it's like, it was in Latin. We used to put these on charts. So para for number of times pregnant, and a number of times having a baby, and grava, number of times pregnant. Just a little side note. All these little things help you because they make the transition. And charting today is undoubtedly different. We did all paper. Okay. Common features. There's a lot of homologous structures. The penis and the clitoris are homologous structures. They have similar types of duties, testes and ovaries. The vas deferens and the ovidum, similar structures. Some of the bulbourethral glands and the Bartholin glands, which are just lubricating fluid during the time of copulation. There's nothing, so we see a lot of these that are play similar roles that are there, or at least what we call homologous or analogous structures. And it underscores the remarkable, for instance, the scrotum and the labia majora. There's so many of these that go along. And I don't know if I have those, but I would be aware of them. And I'd listen to the recording. And on Wednesday, if I can make a note or send it to you, some ideas, I think that would be important. Because I may have one of those, well, which one of these are not matched appropriately together kind of question, that deal. I'm trying to think back and see what else. So I understand there's not a whole, we all start female up until roughly 5 weeks after in gestation. At that point, I think it's the SXY gene begins to demonstrate itself. I'm not sure of the name or the acronym we use for the gene. And then the male begins to develop at that point. That's my recollection. Embryology is, as we speak now, 52 years ago, so I'm a little rough. A little rusty. Gametes, they come together, the hormones you already know about, everything else is accessory. This is up here. So you should know the axis. This is a universal axis. We have a lot of these. This one's as famous as any. This is the hypothalamic pituitary gonadal axis. The hypothalamus is where we produce GnRH, known as gonadotropin releasing hormone. The blood-brain barrier is weakest there. You may recall. And what happens is, and this is obviously pituitary. So typically it senses levels of estrogen, testosterone, and progesterone. You're going to see a nice diagram, better than my horrible drawing. And what that basically signifies is if those levels are running low, the posterior pituitary and the hypothalamus sense it. Gonadic release hormones make things get busy in the anterior pituitary. It releases 2 of its 6 principal hormones, FSH, follicle stimulating hormone, which leads to eggs and sperm, and LH, known as luteinizing hormone, which leads to, depending male or female, testosterone, estrogen, and progesterone. As those levels increase, it feeds back negatively, and as the levels go up, it says we don't need as much gonadotropin releasing hormone. Very standard arc. What's interesting about this system is even though that's standard negative feedback, there are little places where positive feedback takes a role, and none more evident than in areas of female reproductive and particularly the onset of puberty. I mean, that's really when things kind of go, all of a sudden it doesn't make sense. We don't know why. All of a sudden there's a higher threshold of one, or it's less sensitive to something else, and all of a sudden it starts off. And this was sort of one of the center points for my presentation to your colleagues who were here previously, is there's so much stuff going on now in reproduction. Forget the fact that my context of understanding repro, which is not my longest suit, is the fact that my first wife and I are an infertile couple and we adopted. We didn't even have children, actually. We adopted 3 children, okay? Wonderful, I'm joking. So my wife, one of her sons, he and his wife have 1 baby, 1 little girl by IVF. They're expecting another one by IVF in August. My son, again, not biologically related, my eldest, has 2 children, both IVF. There's a lot more infertility going around. We're seeing earlier onset of puberty. We're seeing problems with declining sperm levels, all sorts of stuff. And there's a lot, and I'm not here to answer the question, I'm here to throw it out. It changed a lot. So the early onset of puberty in adolescence, is it a food source? Is it some type of chemicals that were exposed? Is it something environmental? There's a lot of controversy and a lot of speculation about that. And I'm sure there's inevitably there's an adequate number of conspiracy theorists and all that. People who want populations to decline and all that. I don't get into that. But the fact is real that this stuff is going on. That's just a frame to talk about it. So here, just summarizes this GnRH. Hypothalamus reaches anterior pituitary cells, okay, and it says FSH and LH make this. They make the sex hormones, testosterone, estrogen, and they exert negative feedback. When you have particularly increasing sperm levels in storage and byproducts of eggs, of the production of eggs and ovulation, there's another chemical that comes from the gonads called inhibin. And its name tells you what it does, is it inhibits, basically that axis from going into overproduction. And again, the onset of puberty, things kind of go in an odd direction. Here is that, as you can see, all these things. Sex hormones inhibit those target cells that are there, okay? And they feed back on the hypothalamus. Gonads and the ovaries are the target cells. They produce that. And so you look at stimulating versus inhibiting. So these chemicals feed back negatively that are there. This is the oddball. So all of a sudden, and it's coming earlier. I don't know what the current statistics are, but theoretically, the earliest time that reproduction could be achieved, certainly in the female, it's unusual that during the first menstrual cycle or early menstrual cycles, that they could conceive. Okay, perhaps in men it would be you could think it could occur. I don't even think that statistically that is that likely. So even low levels of estrogen, testosterone, progesterone, tiny levels are enough to suppress it. As puberty approaches, it becomes less sensitive. And all of a sudden, not only does it start to release the GnRH, it releases it rapidly. And even then, as it produces more of these hormones, the feedback isn't as effective. It raises the threshold. Oh, you got to produce more to feedback. So they release this in response to those levels. The FSH and LH release more sex hormones. The threshold is raised, so you get more and more, and eventually you get this adult pattern. So it goes from being negligible to that chart that you saw before on my poor diagram. Effectively, try to remember that the idea behind it. We really don't know what signals it, but all of a sudden, it's almost like it becomes much more reminiscent of a positive feedback cycle than a cycle. I'm not doing it. Okay, there's two events in meiosis. It's just busy work. There are two. Mitosis is all about mechanical. Mitosis is how our body basically rebuilds itself. We're hurt. We have to replace something. We make more of the original article and make them exactly. So on the flip side of that, meiosis is all about diversity. Two events. One is called crossover or synapsis. You'll see it in some of the slides. Effectively, we double the chromosomes and then we form something called a tetrad. So these little pairs are called sister chromosomes, one from the male, one from the female, but they're much closer together than this. And what happens is these two guys close together kind of do that kind of thing. They crisscross here and here. It has lots to do with where they're sort of tethered together. Like little protein ball that's in there. And what happens, they will literally exchange genetic information. So this chromosome will go over here, so this chromosome will go over here, etc. And inherently that's diversity. It's called synapsis, crossover. A number of terms. Iasmata. The other one is this. That happens, and remember we have two meioses. I'm not going to include that, but this is called prophase 1. The first prophase is where this occurs. Okay. And the next one right after it is metaphase. Remember they line up, prophase, metaphase, anaphase, telophase, cytokinesis, metaphase down the middle. But you see, it doesn't matter because there's no irregularities in mitosis. Meiosis is called the law of independent assortment. So we have 23. It's not like, oh, chromosome 1, chromosome 2, chromosome 3. Could be any order and facing either way. All moms on this side, all dads on this side. It could be 1, and then 3, or 2, and then 6, and then 8, 10, 11, 12. You got the idea? That's independent assortment. That produces a lot of diversity. Those are the two big events. I don't know if they're on my test, but they're definitely important going forward. That's a SOP question for those kind of standard operating procedure question for a lot of things that are there. The two areas where really we achieve diversity, and it doesn't include just mutations in general. Because reproducing cells is a business where we occasionally errors occur, and sometimes they're helpful, sometimes they're bad. Just depends. So again, reducing the whole idea, these are the sister chromatids, and we call them homologous pair, one member of each pair is from dad, etc., etc. And then this is the crossing over, whatever they want to call it. Remember, those are the individual chromosomes, all 23 of them. Right now, we call that haploid. The half number, they come together for diploid. And just stuff that you should remember going forward. I refuse to rebuild the wheel. I'll go through all these sequence and all the reduction divisions. This is kind of what they're saying with the chiasm. I have a better illustration. So it's sort of like somewhere over here, you can see like these little tips starting to touch. That's where it occurs. You get a little bit of genetic material here, the green from the purple, they do it in color. That's kind of how it works. Done. Don't want to see it. So here, same complement of DNA, same cells here, all about diversity, random alignment. That's the law of independent assortment. A Belgian monk in Prague, Czechoslovakia, discovered this in the 1850s. You all know his name, Gregor Mendel. We named genetics for him, Mendelian genetics. No microscope. I mean, maybe he had a microscope. Yeah, he had a bunch of pea plants and figured all this out. So, I mean, the science is very well understood, because in order to answer why was this happening, all those Punnett squares maybe you drew and things like, they should be ringing a bell for some of you, depending on, how extensive or how unextensive, it doesn't matter your background was. There's some animations here. If you want to look at that, feel free. We just don't have the time. I'm trying to get this all in. That's there, which brings us to testes. Okay. I'm looking at the major anatomy that there. The testes are where I know one thing you should know. Where is it produced? The seminiferous tubules. That's where sperm is produced. The fancy names, I don't think it's on the test, are called the interstitial cells of Leydig, capital Leydig. That's where testosterone is produced. They're all in the same area within these seminiferous tubules. There's some other cells that are in there. Little muscle-like cells, we call myeloid cells, that help to push the sperm out as they begin to be formed. There's some supportive cells that play a role, but those are the two big cells that do stuff that's relevant. And what happens effectively, the sperm are circular haploid cells, for lack of a better way to describe it. There's not a lot to them, but once they get into the epididymis, that's where they are stored until they're ejaculated. That is where they develop and mature. They gain the classic formation of the tail and the mid-piece and the head. And that makes sperm look like sperm, which are readily identifiable. That all occurs in the epididymis. It can store them for a long period of time, not indefinitely. And when they're ejaculated, that's where they come from. Stored there, and then there's a kind of a reverse peristaltic action that rapidly causes them to exit and pick up secretions along the way. Their exit point, and it's a continuous structure. So from once it starts, it continues all the way. The epididymis, the tail of that leads directly into where they're moving, and we named that transitional point the ductus deferens, okay, or vas deferens. Vas would be for blood vessel. That, as that goes on either side of the bladder in the male, it joins with what's called the seminal gland. The seminal gland, with fluid coming out of it, its duct joins with the vas deferens, and it has a new name you're going to see in the illustration. It's called the ejaculatory duct. About 70% of the fluid that we call ejaculant comes from the seminal gland. At that point, now that combination, that ejaculatory duct joins with the urethra as it's exiting the bladder inferiorly, and it joins with it within the confines of the prostate gland. And there it picks up secretions from the prostate called prostatic fluid. They make up roughly the other third or 30% of the fluid. The sperm are immensely new. We're talking hundreds of millions of sperm in a typical ejaculant of what? Half a teaspoon to a teaspoon would be 3 to 5 milliliters. But the fluid is the substantial portion of it that's there. These sperm are extraordinarily tiny, as you might guess. And effectively, once they join with that urethra, we call it the prostatic urethra. You probably had this in anatomy. There's a little intermediate zone. It goes from sort of within the abdominal pelvic cavity to without into the sponge, what we call the spongiform penis. And at that point, the spongiform urethra. Now it's a shared system for reproduction and for urination. So again, the bulbourethral glands are just lubricant. They don't really have a function. We have the Bartholin, capital Bartholin, glands in the female, same function, lubricant. Not a whole lot to say about them. Here's the illustration. So you can see it's rather long. It's about 1.5 feet from where, and it's amazing how much surface area and storage capability there is. This is another common thing that's called a tunica albuginea. It's white, hard, and fibrous. And because these have descended from the peritoneal cavity, there's a coating of peritoneum we call the tunica vaginalis. We see that still in the egg within the abdominal peritoneal cavity, as you'll see when we look at this on Wednesday. So names you should know, tunica albuginea, because it's white and it's hard. It's a hard fibrous tunic that keeps everything inside. And then it's covered because really all of this has descended from in here in the abdominal pelvic cavity. It has a lining layer, they call that the tunica vaginalis. And it's there. And you'll see the structure continues to the epididymis. This is the tail. As soon as it exits that area, it becomes somewhat uniform, about 1.5 feet all along on either side. There's one on the right, one on the left. Here, it begins to widen, and we call this the ejaculatory duct, where the seminal glands and the vas deferens meet. Now they're in the confines of the prostate. The urinary bladder comes in. The urethra comes in, rather. And now we have the prostatic urethra. Here's the intermediate zone, and there's that little bulbourethral gland providing some lubrication, and out it goes, so to speak. These have descended. When they don't descend, and it happens rare, they use the term rare here. I could buy it statistically. Undescended testicles are something that, in a hospital you see that every so often. But if they want to call it rare, that's fine with me. I don't know what the statistics are for today. The whole idea of why testicles descend is fairly straightforward. Okay, it's temperature. It reduces the temperature. Optimal sperm production has to be cooler than body temperature by roughly 3 degrees Celsius. Okay, that optimizes. And it's one of the things they do for infertility. Again, my context is infertility. What they will tell, if they think the male, that maybe if he's wearing briefs, maybe the temperature gradient isn't as effective as you wear boxers. It's one of the first things they would ask you. If they decide, when we had the workup done, I had good sperm count, good motility, and all that stuff. So and statistically, it's interesting. We were told this. It wasn't by just somebody down the street. We went out. We had tried for years. We went to, at that time, the man who was considered the greatest living expert on infertility. He was at the Hospital of the University of Pennsylvania, Philadelphia, at the UP, named Dr. Celso Ramon Garcia in 1980, in an effort to, and we were unsuccessful. Friends of ours who went, they were there for endometriosis. They were successful. I had a child, a little girl biologically. Unfortunately, a lovely lady died relatively young, about 15 years ago. But we were not. And this was all part of the workup that we did. It was all part of all these counts and things like that. That's what they do. So it doesn't matter who you go to. If it's in the cards, it's not. I'm very fatalistic about that. Hey, what are you going to do? And we tried all those strains. We tried in vitro when it was in its early days. In vitro now is an accomplished science. This was, I honestly will tell you, in 1980, you couldn't even talk about it in certain areas. It was like witchcraft or surrogate parenting. Oh, having your baby grow up in somebody else's uterus. Oh my God. I don't know. I don't mean to be nasty about. I don't mean to compromise anybody's values, but honestly, if you're an infertile couple, you'll do anything. Try. You hear these people stealing babies. Nothing like that, obviously, but you'll do anything that you think if there's a way to have that child. And a lot of people, we were fortunate to have, at least to some degree, the financial capability to do this. It's very expensive adopting 3 children in 3 years and 2 weeks. That's why I'm working and I'm going to be 74 in 2 weeks. What's wrong with that picture? Less than 2 weeks. They're still costing me money. Now, this is anatomy, the 2 muscles in the male that cause the testicles to retract when basically if there's excessive cold temperature to basically restore the grade. They're called the dardos and the cremaster. I never remember which is which. So here's the illustration of it. This is the spermatic cord. It descends through an opening in the inguinal ligament. That's a soft tissue ligament, like a tunnel. And this is where, unfortunately, you can get abdominal. This is where you have a rupture or inguinal hernia. That's part of like the small bowel will penetrate through. Extraordinarily painful. The one who's giving me a headache right now has had that repaired. On both sides, having to put mesh in. Not a great idea, by the way. And this is all this blood vessels that are there. This pampiniform plexus that absorbs a lot of the heat that's there. Very, very useful. Here's the vaginalis and the albuginea. Same thing when you get to the female. And here you have the seminiferous tubules. You don't have to go into straight tubes and reedy testes and efferent ductules, but eventually it goes to the epididymis. Cooler, spermatic cord, very sensitive. You can see why all these vital structures are in there. When someone is struck there by intent or by accident or whatever, or there's been some traumatic events like an accident or a fall, that's there. All right, everybody gets the joke, so put up with it. I see this picture, you know what it reminds me of? Ramen noodles. Thank you. God bless you. That's why you came today. Doesn't it? In a perverse way, weird. See, no. My wife will say, did you tell him that same stupid joke? And I'll say, yes. She goes, I know, you're I will not say it. She said, it wasn't even fair yesterday. I got such abuse. Moving on. So they're all, and this is all surface area. All these tubes, it's really just once these are individualized, once they flow together here, this is all like one giant storage mechanism with all, again, folds and twists and compaction. The surface area in a relatively tiny space that's there. Here's the cryptorchism or the undescended testicle nugget, strong association with testicular cancer. Most common cancer, men 15 to 35, thankfully treatable. Eminently treatable. Sometimes even, and it doesn't make someone infertile if they have to remove it, a testicle. The very famous or infamous Tour de France professional cyclist Lance Armstrong had metastatic testicular cancer, probably as a result of this. And yet, no matter what he did illegally, and right, to be able to have had metastatic brain cancer from testicular cancer and have won that race 7 times by whatever means, tells you how extraordinary athlete he is. And I get back to the recurrent point. The problem why men miss things. Men don't have babies, women do. Women are much more conscious of reproductive health, self-breast examinations, going to see the gynecologist. Men don't do self-testicular exams. Forgive me. As long as the material, as long as the equipment works, they're happy. Even if it doesn't work great, they're still happy. They have to be alarmed, and that's what happens. guys. I'm not telling any tales out of school. I tell it to everybody. It's just the way it is. Men are very reluctant to go to the doctor and start talking about ED and those types. It's like, oh my God, the male ego gets in the way. It has forever. Okay. Accessory ducts. This is the epididymis for storage. This is the way it leaves. You heard about the ejaculatory duct and leading into the urethra. So you can see what it's like. 20 feet in length. That's the epididymis for storage and maturity. They are basically, you'll see the illustration, there's not much that they can do, and it has to be contraction in the epididymis to allow this to flow out. The ductus deferens, as you can see, what it leads into is about roughly 1.5 feet, maybe a little bit less. Okay, join the ejaculatory duct. Vasectomies are very effective. Again, they do not impair function. Here's another family one like this. My wife has 5 children. The older 3, last one in 1985, 6 years later, 1991, my wife's ex had a vasectomy that they decided to have 2 more kids and did. They wanted to have a girl. They had 4 boys and a girl. That was post reversal of a vasectomy. I'm like a medical history. Everybody is. As life goes on, you wouldn't want my mom used to say, if everybody threw their problems into the middle of the room, you would take yours back. It's true. She said that when my dad was dying and didn't survive heart surgery. We were waiting to see if the next surgery worked, and it didn't. And she's hearing the other people talking about their problems. This is anatomy. Again, the big deal is here. The erectile bodies, the spongy portion of the urethra that are here. This is the spongy. There are 2. These are corpora, which means body. The cavernous ones are really the ones that are the bulk of the erection. The spongiform one here, which is singular, surrounds the urethra, and really its form of the urethra in the middle keeps it open, so it's sperm are allowed to pass. And then there are mechanisms farther back within the system that prevent urine from being incorporated with the ejaculate. So it's a pretty interesting way that all takes care of. That is the external part. Foreskin, you don't see. Most men in this country that at least I've seen are circumcised. That number is a little bit low. The risk of reduction for HIV is very significant, as well as other infections. So you don't see it as much in other countries. The problem is that foreskin is relatively delicate. It tears, and it's easy to get infection or debris that's in there. They didn't give me a choice, 8 days. In Judaism, it's called a bris. It's the ritual circumcision. Blame Abraham. You remember him? Old guy, son, sacrifice, maybe not, angel. Right? Bible stuff, Old Testament stuff. Thank you. But Jewish men are so angry. Look what you did to me. 8 days. So here's the erectile tissue. That's fine. And yeah, it fills with blood. And because when it fills, it engorges, but that also effectively makes the venous drainage impaired until the stimulation wears down. It's all because of something called nitric oxide. That's where ED medicines take their point. Arousal, in both the male and the female, releases nitric oxide by way of the parasympathetic nervous system. Remember, arousal is parasympathetic. Okay. On the other hand, climax or ejaculation is sympathetic. The parasympathetic nerves in that area, the sacral ones, stimulate the release of this nitric oxide, causes the structures to engorge with blood. As long as the nitric oxide there, they stay engorged. So what are ED medicines, which extraordinarily used today, because there's so much of this dysfunction, it makes you wonder. But what they do is they basically prolong the actions of nitric oxide. They inhibit the metabolic pathway that breaks it down. When you take a medicine, your body breaks it down. This blocks strategically the pathway. So all of them work the same way. They're on this. They may work at different parts of the pathway. All these meds you see advertised freaking everywhere. And they became very affordable and all that stuff. Everything. Everybody wants a piece of that particular. This is a commonly used thing. This is anatomy, the perineum, all the structures. And we look at the male and look at the female. So pubic symphysis, ischial tuberosity, and the coccyx in the rear. Effectively, this is where the structure. You'll see in the female, it's very, very similar. If you think of what happens with gender reassignment surgeries, I mean, it makes it eminently clear how things could structurally be altered if that's what the individual felt was the appropriate approach for them. That was there. The seminal glands, these are the first part of physiology, I think, because what it tells us is that they're on the back of the bladder. It's just like the stomach, it's pH. Vaginal fluids in the reproductive female are acidic. They're guarded by acidity made by something called lactobacillus. That's the major protection against infection, against disease. It's a highly acidic environment. You have to neutralize the acid. That's the alkaline. This really neutralizes it. So it's very much about, look at that process. And I don't generally do it. I mean, it's very, very complex to see all the different steps. Fructose is the only sugar that activates sperm. Citric acid helps to preserve it. The coagulating enzyme prevents it from outflowing. So it coagulates it as it enters so it doesn't flow back out anatomically. And prostaglandins activate a lot of the different mechanisms to allow things to flow, to relax structures, to allow flow. There's a lot that's involved. And this is 70% of the volume. And when you see on crime shows, when they get an ultraviolet light, they're looking for, it fluoresces yellow. So when they start looking for, there's fluids here, whether they're looking for a rape or, and hopefully not, those things you just sad when somebody's been physically assaulted and terrible. The prostate's a little bit different. It is about 30%. It's not exactly clear. The acidity isn't enough to offset the alkalinity. Yeah, the citrate is a preservative. It's that white milky consistency that's so evident on sperm. It plays a role in activation. It's this. In males, the way you determine prostate disease, and it's just like breast cancer, it's the number one cancer in men statistically, a very, very high percentage men get prostatic cancer. The way it's called a digital rectal exam. It's done through the rectum by whoever is the examiner, the physician, or whomever would do it, the practitioner. And effectively, they're able to feel if there's hard spots or irregularities. That's part of it. Now we have this PSA, and it will measure if it's high, it strongly suggests problems. Also, it monitors during treating for prostate cancer. The treatment, you monitor it by doing PSA every year and a half. Whenever I have my annual physical, they do a PSA. They do one of those tests. Not fun, but hey, goes with the territory. And again, just lubrication more than anything else. This is I did not have prostate cancer back in October of 21. I would say the vast majority of my prostate removed because it was so enlarged that it was obscuring. And that was from treatment for my bladder cancer. So it was a byproduct of what had gone on previously. My doctor removed it, and they used a laser treatment to remove it. It worked very well. I mean, no problems. The recovery from that's not pleasant. But at that point, I'm 70 years old. So and they have some good medicine. We use a thing called Flomax. A lot of men my age use that because it's irrelevant, even if they don't have problems. So I had this transurethral resection that was done. Prostatitis is an infective problem, generally not passed on to partners. Most men experience it once in a while. If it causes death, it's because someone ignored it. This is something that does not kill rapidly. It's very rare in prostate cancer. I'm not saying it doesn't, but it's very rare. When they talk about this, to me, it's like it's a diagnosis where someone has not treated it. The average time people will have be a lifespan after diagnosis of 10 to 15 years. It's not quick. Usually they've died of other factors, and that's just another diagnosis. Sort of like everybody dying of COVID when they're 100 years old. Oh, but they tested positive for COVID. It must be a COVID death, right? It's 100. They're gonna die. Ooh. So all of that type of stuff. Here's the numbers, the millions of sperm that are there. Again, fructose is to activate. That's for ATP because it's the only thing that they can use. Activating the sperm, movement, neutralizes the acidity both within the male urethra and the vagina. And all of these things, decreasing viscosity, mucus is still within the cervical area, reverse peristalsis, basically helping the sperm to ascend, suppressing immunological responses, etc. The clotting factors issue. There's very little here in this. And so in the 10 minutes we have, what does it do? Again, this is the state of arousal is parasympathetic. We did this in AP1, the autonomic nervous system. The climax, orgasm, ejaculation in the male, that end event is sympathetic. They work together. Here's the nitric oxide part of it I talked about. Engorges, venous drainage can occur. So as long as it's got nitric oxide, or the stimulation is there, and then once it concludes, and we know all the different ones that are there, and these things are so, a lot of them, I mean, there's some things that, there's many things that cause it to occur that inhibit it. What people have dysfunction is not always a physical problem or nitric oxide or aging. A lot of times there's emotional, psychological problems. There's so many, it's not clearly understood. And then it propels itself out by this wave of sympathetic nerves. And we have a similar sort of reflex contraction in the female for climax. And obviously in the male, in order to have fertilization, we have to have a climax or ejaculation in the male, not so with the female. Okay, and then you can see it expels out at a relatively rapid rate. Okay, and that is considered the climax or orgasm, and the usual sympathetic rings, heart rate, blood pressure that people experience. The interesting thing in women, we don't see this refractory period, or it's very minimal. In men, it varies. As you get older, there's a longer refractory period. The young individual, sometimes it's very minimal. I can't give you, exactly, the factors that influence it. It seems to be very individual. And that refractory period we call resolution, where everything seems to resolve by itself because the nitric oxide, and they should have a capital O there. And as you can see, over age 40, that's a big number, I think, compared to what it might have been. Again, back in our day, most the dads of my generation were all in World War II. Nobody's talking about this stuff at the time. They're not. It's very, very different today. Very, very different today. These things. And so there's a lot of different problems that could be with it. Diabetes, a big player. That is there. This is the interesting, and I'll sort of summarize it here as we conclude. Because that's all the thing where the sperm are growing. The only thing you really have to know about this is, we call this the acrosome. This has enzymes to get through the shell of the egg. The genetic material is all up here, the haploid. It's there. The mid piece is all about energy production. It contributes nothing to fertilization. And the long tail is just this, a long tail for motivation. And again, side to side, just what they look like, micrographically. And so this is how they come into the epididymis, and this is how they go out. That's their better part. What's that, 10 weeks at least? I'm not going into the compartments. This is significant. Sperm are produced after roughly age, at whatever they start production, 11, 12, 13, 14, whatever they are. That's after that non-self period of immune system. If they get into your bloodstream or into other tissues, your immune system attacks them. They're foreign. It's just it's a barrier. And so in cases of trauma or infection, maybe surgical intervention that didn't work out the way it was supposed to, you can cause a major immunological phenomenon. And whenever there's trauma to that area, an auto accident, I mean, these are the kind of things you're going through the back of your head. Do we have this problem? It's there. I don't do anything with that. 1 in 7 couples. I think that, and I think it's greater, to tell you the truth. Gradual decline over the last 50 years. All of the possible culprits. Interesting area of study for environmental toxins. More that are there. Here's the axis. We're almost done. And lastly, this part of the chart. We know prior to birth, this is part of the process of basically finalizing development. Maybe this little spike after birth, maybe the complete descent of the testicles, maybe finalizing the fact that the barrier has been created or early start to function. I don't know, speculative. But after that, once male hits puberty, it ramps up. Depending on who you read, in a couple of years to maximum, and stays that way. There's plenty of evidence of men without the aid of today's IVF, things like that, in their 70s, 80s, or even 90s, biologically, without IVF intervention or that type of stuff, being able to sire children. Exactly how the quality of the sperm is something people have taken into question as well. But you can see about 3 years to achieve it. We know about secondary sex. You can read that as well as I can read it to you, in men and in women, and that's it. Thanks, everybody. We'll do female. It's probably I'll never get it all done in one day. We'll have to conclude it on Friday. I just know. I hope that helped.